Hi guys, Miss Francis here, and I'm here to finish up our unit on biochemistry and organic molecules. The last macromolecule that we're going to discuss is nucleic acids. You guys are familiar with nucleic acids. You've discussed them back in living environment. The two types of nucleic acids are ribonucleic acid, RNA, and deoxyribonucleic acid, DNA, where adenosine triphosphate, or ATP, is also categorized as a nucleic acid because it's a building block for RNA. So DNA provides directions for its own replication. It's the blueprint. Therefore, it also directs RNA synthesis, and that RNA synthesis is going to control the protein synthesis, because the RNA ultimately determines the polypeptides, and we all know polypeptides combine to form proteins, where ATP is going to be the energy that's used by the cell. So let's go through what a nucleic acid consists of. So nucleotides are the building blocks of nucleic acids, and each nucleotide consists of three parts. I like to say it's the house, the pool, and the garage, where the house is the sugar. In DNA, that sugar is deoxyribose. In RNA, that sugar is ribose, the phosphate group, and your nitrogenous base. Now, the phosphate group is negatively charged, which is going to be important when we go to visit the DNA Learning Center and we do gel electrophoresis. You'll see that the DNA is going to travel down towards the positive side of our gel. Now, these nitrogenous bases, which are one component of our nucleic acid, can be categorized as pyrimidines or purines. Your pyrimidines have one ring, whereas your purines are going to have two rings. And the way I remember it is thi, si, and u. So thymine, cytosine, and uracil are pyrimidines. And down here, you'll notice that deoxyribose is the sugar in DNA. Ribose is the sugar in RNA. Because if you notice, there is one oxygen that's missing. So that's where you get the deoxy name from. So here's just what I just said, that the nitrogenous bases or nitrogen bases come in two types, purines and pyrimidines. Pyrimidines have a single ring and purines have a double ring. Again, the sugar can vary. It will be ribose in RNA and deoxyribose in DNA. Now, on the AP exam, you'll never be asked to draw ribose. On the IB exam, you could be asked to draw ribose, but in any case, you should be able to recognize the shape of ribose. Ribose is a sugar. It's a pentose sugar because it's 5 sides. Just like any other macromolecule, nucleic acids are synthesized via dehydration synthesis. I'm going to connect one sugar in one nucleotide with the phosphate of the next nucleotide via a phosphodiester link. This is going to create what we call the backbone. So you have a phosphate sugar backbone. So here is my phosphate sugar backbone, where these bonds between my phosphates and my sugars are my phosphodiester bonds. If I were to circle an entire nucleotide, I would include my phosphate, my sugar, and my nitrogenous base. Now, DNA and RNA have some differences. RNA is a single helix, whereas DNA is a double helix. And this was first discovered by Watson and Crick. Here's a nice video that discusses the structure of DNA and RNA. And now I want to go through some similarities between DNA and RNA. Well, both are made up of chains of nucleotides, each of which has a phosphate, a pentose sugar, and a nitrogenous base. Both contain nitrogenous bases adenine, guanine, and cytosine. Both contain a sugar phosphate backbone, and both are negatively charged because they have a phosphate group. So in DNA, you're going to find 2 strands. RNA, you're going to find 1 strand. The nitrogenous bases in DNA are adenine, thymine, cytosine, and guanine, whereas your bases in RNA are adenine, uracil, cytosine, and guanine. The pentose sugar, this is deoxyribose, whereas in RNA, it's going to be ribose. The function of DNA is to code for RNA, and the function for RNA is going to code for your polypeptides or your proteins. So the location in eukaryotic cells for DNA is going to be the nucleus, the mitochondria, as well as the chloroplasts, whereas the location for RNA is the nucleus and the cytoplasm. So what I would like to review with you is just the structure of DNA, where we say that DNA has a sugar phosphate backbone and 3 ring rungs. So the backbone is made out of sugar phosphate, and the rungs are your nitrogenous bases. Here's a nice little video, again, that summarizes DNA and RNA structure. And here's a nice video that summarizes the differences between DNA and RNA. So this concludes our biochem and organic molecule unit, and I hope you found this helpful.